and thank you very much for coming. It's a great pleasure to be here today. So what I wanted to talk to you about today is some of the work that we've been doing in the past 10, 15 years, I'd say, in the area of synthetic, nowadays known as engineering biology, in particular on how we can use biology to design controllers. So how we design feedback control loops inside biology. Now, when I started working in this field, we were looking at how you can build and design new biological systems by following an engineering, design, build, learn, test cycle like everybody else in the field. So there you'd have like design objectives and specifications, and then you try to design according to those specifications. And then you pick now biological parts, not like electrical parts that you do in electrical engineering design, for example. So you build promoters, you pick promoters and genes, put them together, run simulations, and then go in the lab and assemble the DNA, synthesize it, and then try to see whether it works the way it was intended to do. And that's where the feedback loop, these design builders, test cycle, usually fail. So in the first few years of doing these, we weren't able to get this to work. And when we did manage to get it to work, and we tried to move our designs into a different kind of system, our designs wouldn't work as intended, especially in different environments, change the temperature a bit, and the performance was going down. Now, we were, as engineers, we weren't very happy about that, and obviously the designs we were having were missing one thing. They couldn't self-adapt, so you'd expect that in a new environment there would be an opportunity for the design to adapt to the new environment. We're wondering whether feedback control, so adding feedback to these devices, to these systems, would actually help. So we embarked in a systematic approach for designing biocontrollers. But for those of you that it's been a while since doing engineering, I mean, in terms of feedback in engineering systems, I mean, it's everywhere, and all the systems work seamlessly just because the feedback design in them allows them to work so perfectly. So thermostats, for example, you set the temperature, and the temperature gets to that level. Cruise control is the example that we nowadays give. So if someone asks, what is feedback control? Well, you run your car at a certain speed. You want it to go to 60 miles an hour. It measures the difference between the speed that you're doing versus what the desired speed you want to have. And that difference goes into a controller that automatically sets the throttle to the car. And I'm sure we've all tried this, at least the one on the left trying to go uphill. It doesn't, up the hill, the car will slowly adapt its speed in order to go to the right desired speed. I'm sure I hope you haven't tried the one on the very right. No tuning, no rebuilding, it just works. Now, natural biological systems, the cells in our body, use feedback extensively. So perhaps my very first experience with feedback was actually a project with Judy Armitage, who is in the audience, on looking at chemotaxis. So this is bacteria experiencing a grain of sugar, and they do a biased random walk, trying to figure out where to go, but they all eventually congregate around the sugar granule because they want to be fed. And what happens here is that inside each of these bags, there is a sensory mechanism whereby when the ligand is bound to the receptor, there is a signaling cascade down to the motor that tells the motor to to turn clockwise or anti-clockwise, or the flagella open up and close, to create this biased random walk. But then, at the same time, there's a feedback mechanism to reset the receptor so that it can actually sense other gradients as and when they arise. And what happens in Berg's experiments, which were quite influential in the field, what happens is that if you add an attractant step, for example, here, if you step up of food, then there is an activity inside the cell of phosphorylation down the motor, phosphorylated key Y. But then that readapts to where it was before, so that the cell is ready to sense again. And that is adaptation at the level of, perfect adaptation at the level of signaling that allows this sensor to work reliably. Think about having a thermometer that has the same sensitivity at very low temperatures, but also at very high temperatures. So why can't we do this inside synthetic biology? Why can't we do something similar for synthetic biology? And that's the question we asked about 10 years ago. And I'll tell you a story, but it's not linear in as much as research, but I'll try to put it in a sequence that makes sense. So the first thing we started thinking about is feedback in a synthetic sense has happened, right? These are the very first designs, and Jim Collins's toggle switches features here as well. I mean, these are positive and negative feedback loops. These are the very first synthetic biology constructs that appeared in the literature. And all of them have feedback. So the first one is negative autoregulation around the gene. So you just put negative feedback around yourself. Here's a toggle switch, and all these are oscillators with sequences of repression. So it was possible to do it. So why not be able to actually design it the right way? Well, in order to build a feedback circuit, what we need is to design this grayed out box. So we need a way of comparing the output of the process with the input. So that's this comparator, this hammer interaction there. And then with the error, if you want, is to go into a controller circuit. But this is a cartoon, right? In biology, you can't just put negative, you can't just put summing junctions, right? But here's a way that you can compare signals. Let's say the process takes molecule U to molecule Y, and I have a molecule Z that annihilates with molecule U, so it takes it away. So if I crank up Z, U will go down and Y will go down. So eventually, I'm getting what I need. And actually, this kind of annihilation, molecular sequestration, is what is known, what has been done in the negative autoregulation signal circuit I showed before. But it can happen at other levels as well. So at protein levels, you've got sigma factors annihilating. And at the RNA level happens with small RNA annihilating with messenger RNA. And that's the kind of circuits we've been building for the past few years. So I want to spend a little bit more time on that. So how do you close the loop for this? In this case? Well, you just make sure that molecule Y cranks up the production molecule Z. Because if U is high, Y is high, Z is high, U will annihilate with Z, U will go down. So that's a negative feedback. It's not a traditional negative feedback you see we teach, but it is a negative feedback. And our negative feedback circuit look like this. So the input comes in, produces the mRNA of this gene and the protein. The protein is a transcription factor to produce a small RNA that annihilates with the messenger RNA of this output, so thereby pushing it down. So I'll explain this again in a different way in a minute. But let's look at this annihilation reaction first. So you have molecule, an input at the very top, U1, producing a messenger RNA and a protein. And what you're doing is you're designing a silencing, a small RNA to annihilate with the target mRNA. So this is essentially in block diagram terms what you're doing with the sRNA. Only the net, what remains, will actually be translated into protein. And you can tune this annihilation by designing the sRNA the right way. Okay, so this is something we have control to do. So, and we can build models for that, mathematical descriptions, and tune parameters and choose this in the right design, build, test, learn cycle. And this is what we did in this project. So here's the feedback circuit that I'm building now in block diagram terms. You've got an input producing an mRNA, but only the net mRNA produces the protein X, because protein X is also producing its own sRNA. So that's where the loop is closed. And we get, so this is a simple model of how this circuit works from the input and the disturbance U2, and you get the right kind of response. So you are trying to now start thinking about how do I design this circuit, and you can think about ways of picking your promoters. So this is now the question of engineering. How do I design these these transcription circuits to get the desired response, because you want to have X to follow U1 but reject U2. And now if all the processes were linear, so this is now what we teach in our undergrad. So this is block diagrams with transfer functions, blah, blah, blah. What you get at the end is you need to get G1 to be high compared to G2, because that means that Y follows U rather than U1 rather than U2. And lo and behold, we go out and pick the right promoters. And this is what the electrical analog of a synthetic biological circuit looks like. So this is a plasmid, as it's called, with all the promoters, with all the genes that need to be introduced in the right place in order to get a circuit that mimics the circuit above. And these are the types of experimental data you get. So the input here is the input on the left, on the bottom axis. So that's U1. So as U1 is increased, you get more and more of the output. But every 4 of them correspond to different levels of U2. And obviously, if U2 is 0, then you get no feedback. So that's the open loop circuit. So you can see the open loop circuit has higher variability. As you crank up the gain, you get more robustness to that level of U2. So our circuit achieves the steady state response we want to get. And you can then go on and do more analysis and more experiments. For example, time series experiments in order to see the time response. So this is the time response experiments done by Scott. So using one of the Kai-Bio devices, actually Harry designed. So you've got, for example, the open loop circuit being more slow, but producing a higher output compared to the closed loop circuit, which is faster, but producing a lower output when it has a little bit of overshoot, right? So this is what we expect to see in closed loop designs. And you can do more experiments like this to characterize these types of systems. Now, it turns out that this very simple annihilation reaction I mentioned before, so when you have two molecules annihilating with each other, is a very important, actually, feedback loop. It does more than just close the loop. It looks like an integrator in the ideal case. So that does integral feedback, not just feedback. And so we asked the question whether we can do more than proportional integral feedback. And then, so my former student, Manos Alexis, he looked at differentiators, circuits that take an input, and one of the molecules actually differentiating the signal. So if you have a sinusoid, will it follow a sinusoid? And this actually circuit, which looks quite complicated, does implement a differentiation for the input U. And together with the integrator and a proportional, you can build a PID controller if you wanted, whereby you could actually tune some of these parameters the right way. This is too complicated to implement in the lab yet, but in simulation, what you get is what you expect. So a badly tuned PI controller would have these undulations, but a good design of different types gives you this damping that you need in your designs. Now, as I told you in the beginning, research is not linear, and that was a very linear story. When we started looking at feedback control, I started with a PhD student, actually, with Judy, 15 years ago now, Mark Chung. We started looking at these signaling cascades and how we could do feedback in signaling pathways. And in signaling pathways, it doesn't make sense to have annihilation between two molecules. What you have there is proteins, let's say, in state 1, unphosphorylated, and in state 2, phosphorylated. And what happens is that you want to shift the balance between being unphosphorylated and phosphorylated by pushing left and right with these inputs, U1 and U2. Okay? So this is a different type of feedback mechanism, and I'll explain why I think why this is the case to the annihilation I mentioned before. But it is a feedback mechanism in that, for example, for the circuit we build with integrated excisionase circuits, so this is, let me explain what's going on here. So these are flipases that flip DNA. And you can have DNA pointing this way, in which case the promoter is here, you get nothing red. But in the interaction of one of these integrases, this piece of DNA flips this way. So suddenly now you are getting to read the GFP. But you also read the excisionase, which flips it back. So you can now tune the rate of flipping left and right. And so it's exactly this type of system where you can push from one way to the other. And once you close the loop in this way, you get populations that are narrower. So these are distributions of cells with this green fluorescent protein, so that the closed loop is narrower and more repeatable. And this is a clear indication of the presence of negative feedback. But the fundamental difference between this approach of comparing signals, which is a molecular sequestration mechanism, and the approach that I just described with pushing left-right, is that the second approach does a signal sequestration. So in the second approach that I mentioned before, you have a molecule U producing a molecule Y with a certain flux. And what you're doing is you're introducing a molecule Z that diverts the flux into another direction. And the loop is closed by having molecule Y cranking up the flux that goes in a separate direction. And this is a feedback loop as much as anything else, because U is high makes Y high, makes the speed away from U to Y high, which means less goes from U to Y. So it steals away some of the flux in different directions. This kind of approach is actually quite prevalent in most networks that have to do with metabolism or signal transduction. And this is the approach that we used in the feedback circuit, 10 years ago, 15 years ago with Mark Chang. So let me show you the approach we follow. So you've got the input signal going through this phosphorylation cascade, just in much the same way as in chemotaxis, we have this histidine kinase getting phosphorylated, which phosphorylates a response regulator, which produces an output. So this little yellow blob means that the protein is phosphorylated. And what Mark did is he cranked up this backward arrow here. So he removed the flux of phosphorylation by adding an extra phosphatase. But what you could also do quite easily is steal the flux from the very top. So you can add a spurious response regulator that also responds to the same histidine kinase, but doesn't actually go on to do anything else. Okay, so that's now siphons away some of the histidine kinase, histidine kinase in a different direction. And of course, the way you'd close the loop here is you'd produce more of this protein. So then when the flux goes high on this direction, you produce more of your secondary response regulator, and it steals away. This is a negative feedback loop, and actually very easy to implement. It shapes the signal response much like every other feedback control circuit. It reduces noise. It implements a robust controller. And it's the natural feedback choice for metabolic and signal transduction networks. It also does something else we're very keen to achieve in synthetic biology, to reduce crosstalk. And what I mean by that, I think actually when we got the experimental results from here, even without input signal, what we're getting is a high output. And I was like, how can that happen? It turns out that there were other histidine kinases in the system that were phosphorylating our response regulators. So there were X's floating around that were actually producing some of the output without any input signal from us. Now, adding the feedback circuit there gave us the right response in that now we became insensitive to the molecule X. And so we could get a response in the output, even in the presence of the crosstalk signal, which was quite interesting. Now, I talked about feedback within cells. Right? So these are signal circuits that you can go inside cells and rewire pieces of DNA to close the loop around processes you want to control. But cells, you can have feedback at different levels. You can have feedback between cells. So you can have cell type A talking to cell type B, and cell type A being the controller. And cell type A and cell type B can be different cells, different cell types. They could be mammalian cells and bacteria. You could also have what Harry alluded to. You could have cells which have very, well, say, simple biology, like optogenetic, or responding to optogenetic signals, in feedback with computers. And that's actually amazing. So when Harry produced this response, so this is a response to a set point on a bulk population inside this Chi-Bio device using a PID controller in the loop with optogenetic control, that kind of thing was impossible to do in implement in this way, inside cell, in situ. But by linking a computer around, this was very easy and very possible. But this now creates a map of feedback approaches around biology, where you can have them inside cells, between cells and the environment, between cells and computers, between cells of different types. There may be plants, there may be mammalian cells, cells of different cell types talking to one another. So we've teamed up with a number of, with collaborators at Bristol and Imperial, and we're now embarking on a 6-year program grant funded by EPSRC on essentially building the theory, software, wetware, and hardware that will build biocontrollers for engineering biology. And we're looking at applications in the environment, in biomedicine. This is work with Wei Huang, sitting over there, and in agriculture. So I'll talk a little bit about in the next slide, because I think that will tie together, hopefully, some of the pictures that you can see below. But obviously, this is a technology that is key for the future of society. But it needs to be developed in a responsible way, and I think that's something we also are looking at to explore in this program. Now, agriculture. I mean, feedback in agriculture is quite important, even at this level of growing plants. And if you're a keen gardener, you know that when you're growing peas, you don't need to fertilize your soil, because inside the soil, there's these bacteria that like to come in and colonize plants. And actually, if you look at the roots of peas, they have these nodules, and what they do is they infect the plant, and they go inside these nodules and create and fix nitrogen, give fertilizer to the plant. And Caroline has been looking at these designs. So this is an interaction between a bacterium and a plant in feedback, because there's a sense and respond mechanism to change its metabolic state in order to come to produce the ammonia it needs, release the ammonia that it needs to the plant. But you can't do this for all plants. And of course, you know, peas have evolved this mechanism, and other legumes have evolved this mechanism that don't need fertilizers. But that plants we care about, like for example barley, they don't have this mechanism. So the question is, can we think about having a community of cells, for example, that come together and produce and provide these services to the plant, so come close to the roots and fix nitrogen for the plant so that it doesn't require excessive amounts of fertilizer in order to grow. And this is work that, again, is part of this program grant and work done by Bertha and Caroline in collaboration with the Field Pool Lab. So with this, I want to finish and thank members of the group that have worked throughout the years in this direction, as well as collaborators in Oxford and beyond. So whoever is italic are the biologists in the group that are not control experts. Whoever is underlined is the opposite, so they're control experts but don't know much about biology. Whoever is upright is exactly people that can go in the lab, come back, analyze data, understand the control behind, and going back and do more designs. So with that, thanks to the funders again. Thank you for listening.